Hello students, welcome to EG Pathshala. I am Farhan Jalis Ahmed from Hamdard University, and today we are going to discuss about Introduction of Microspheres. What are microspheres? From paper Novel Drug Leaf System Part 2. Now in this module we will talk about the general information about microspheres. What are microspheres? Their advantages, their limitations, then various components of microspheres which basically form a microspheres and different types of microspheres. Now first of all we should understand what is a microsphere. As the name says, it's a combination of two words, micro plus sphere. So micro means anything which is in micron size, sphere means which is spherical in size. So it is a spherical empty particle. Now the main objective of designing a microsphere is to protect the drug molecule from external environment, to control the release of the drug medicament from the matrix, and microspheres are basically free-flowing powders. They are dry, free-flowing powders, and they mostly consist of polymers, synthetic polymers, natural polymers, or proteins, which may be biodegradable, which may not be biodegradable, and they are having a particle size which is around 200 micrometer or less than 200 micrometer. And they are made up of polymeric substances in which the drug is basically either it is dispersed throughout the matrix or it is encapsulated inside the matrix. Now if you compare microspheres to microcapsules as clearly shown in the figure, microcapsules are they may be spherical or they may be irregular in shape, and in this case the drug is basically encapsulated inside a coating, while in case of microspheres they are essentially spherical, and in this case drug is uniformly distributed throughout the matrix, either in the form of spheres or in the form of particles. It is embedded in the or entrapped in the matrix, which is maybe a polymer, it can be a protein. Now microspheres, basically the proteins which we are using to make a microspheres, we can use albumin, gelatin, collagen, carbohydrates, agarose, carrageenan, chitosan, starch, or chemically modified carbohydrates like polyacrylyl like dextrans, polyacrylyl starch. So these are the things which are used as a natural polymer in the preparation of microspheres. Now in the rationale why we have to develop microspheres, microspheres basically they can give us a very accurate delivery, because in this case drug particles are coated or maybe entrapped or dispersed in the polymer matrix. Polymer matrix control the release of drug from the sphere, and because they are spherically sized particles of micron size, their surface area is also very large. Now if you have monomer dispersed particles, the release rate will be very accurate as per the desire or as per the control by a scientist. So if you want to release in 12 hours, you can achieve a release in 12 hours. If you want to release in 24 hours, you can achieve a release in 24 hours. So that is why they are ideal for giving accurate delivery of drug inside the human system. Then second advantage or rationale is that they provide a very good protection. The drug molecules which are sensitive in nature, they may be sensitive to atmospheric oxygen, they can be sensitive to acidic environment of stomach, they can be sensitive to enteric or alkaline environment of intestine. They can be protected by giving a protective coating by a inert polymer. So in that case, when drug is entrapped inside the microsphere, it is not exposed to environmental oxygen. It will not come in contact with the acidic media of stomach. It will also not come in contact with if it is coated with enteric polymer, alkaline media of small intestine. So this type of protection we can give by designing a microsphere. Apart from this, we can also use it for protection of the GIT, because those drugs which are highly acidic or drugs which can irritate the gastric mucosa, those drugs when they are coated in a polymeric coat, they are not directly coming in contact with the gastric mucosa. So they are not irritating the gastric mucosa. So that protection you can provide to the drug or you can provide to your GIT lining. Then we can control the release, as I have said earlier, accurate delivery, we can control the release according to our requirement. Whether we want first order release, we want zero order release, those can be used by manipulating the properties of polymer. Then finally, the main rationale of designing a microsphere is to improve the patient compliance. So how patient compliance is improved? Because when we are converting a multiple dosing drug into a single dosing drug, or which can be given once in 24 hours, the patient compliance is improved. Because by designing a controlled system, we can control the release throughout the day by using a controlled release polymer. and drug will be released throughout the day inside the GIT, and the patient has not to take maybe dosing 3 times a day or 4 times a day. So he will take only once a day. So patient compliance is also improved. And secondly, when by controlling the release, we are also reducing the side effects. So when side effects are also reduced, it will also indirectly improve the patient compliance. Now the ideal qualities of a material which can be used as a microparticulate carrier. First quality is it should be able to provide a longer duration of action to the drug which is being encapsulated. It means it should control the release of the drug through its barrier. Then by controlling the release, we should be able to increase the therapeutic efficiency of the drug. Then the control over the content release should also be easier. You can easily control the release according to the requirement of the drug, according to the requirement of the disease, and according to the requirement of the patient. Then drug should also be protected against the atmospheric oxygen, it should be protected against the acidic environment, it should be protected against the alkaline environment, wherever the drug is sensitive to get degraded. Then it should also be possible that we can reduce the toxicity. First thing, the carrier should be non-toxic in nature, and secondly, it should also reduce the side effects by controlling the release. Then the material which we are using as a carrier, it should be biocompatible, it should be inert, it should be non-toxic, it should be stable, and preferably it should be water dispersible or water soluble, so it can be given by oral route. Now in the figure you can see different types of polymers which can be used for the delivery of or for the design of microspheres. These polymers can be classified as synthetic polymers or natural polymers. In synthetic polymers we have further classification. It can be biodegradable or it can be non-biodegradable. In non-biodegradable we have polymethyl methacrylates, which is known as PMMA, we have acrolein, and we have epoxy polymers. These polymers are used where biodegradability is not required and the polymer can be exited from the body through GIT. Then biodegradable polymers, we have lactides, we have glycolides, which is known as PLGA, we have its copolymers, we have polyalkyl cyanoacrylates and polyanhydrides. So all these are biodegradable, and they are basically used in those places where it is not supposed to come out from the body, and it will biodegrade there naturally over a period of time, forming carbon dioxide and water under natural biodegradable mechanism. Then we have natural materials like proteins, carbohydrates, chemically modified carbohydrates. Amongst proteins we have albumin, gelatin and collagen. In carbohydrates we have starch agarose, we have carrageenan and chitosan. And in chemically modified carbohydrates we have the example of polyacyryl dextran and polyacyryl starch. So all these natural polymers are also very commonly used. The difference between natural material and synthetic material obviously is natural materials is from natural origin and synthetic material is from polymeric origin. But the mainly in some places we are using natural polymers, but if you want a proper control release of the drug from the microspheres, then mostly we are using synthetic polymers. Now let's talk about different types of microspheres. The first class which comes under microspheres are types of bioadhesive microspheres. These types of microspheres show a prolonged residence time at the site of action and causes close contact with the absorption site and produces improved therapeutic action. They are prepared by using bioadhesive polymers, and bioadhesive polymers by different mechanisms they can adhere to the biological membrane, and by adhering to the biological membrane they slow down their eviction or their emptying from the site of release. So when they stay there on the site of release, there they release the drug and drug will be absorbed from the site of absorption. So bioadhesive microspheres are used basically where drug is supposed to be absorbed from a specific site, either because of absorption or because of protection. Then we have second class is magnetic microspheres. Magnetic microspheres are supramolecular magnetic particles that are small size, micron size, to circulate through the capillaries without causing any embolic occlusion. So it means their size has to be less than 5 micron. But they are adequately susceptible, they are ferromagnetic in nature, and they can be taken into microvessels and then hold into the adjoining tissues by applying an external magnetic field of about 0.5 to 0.8 Tesla. So these magnetic field can be applied externally by placing a magnet over the skin, and the microparticles will be attracted towards the magnetic site, and there they will be accumulated. So we can have site specific delivery by using these magnetic microspheres. Now let's talk about the different types of microspheres. Microspheres can be of depending upon their physical properties, they can be floating microspheres, they can be radioactive microspheres, there are polymeric microspheres. Floating microspheres are gastrointestinal in nature, and they are made floating because they our scientists try to increase the resistance time in the stomach, and they are having low density system, and they have adequate buoyancy so they can float over the gastric contents and stay in the stomach for longer period of time, because they can evade the gastric emptying because they are floating on the upper surface. So they are not emptied from the stomach, they stay inside the stomach for longer than 3 hours, and drug is released slowly. So they are basically designed for those drugs which are absorbed from upper part of the GIT or from stomach. So that is why they are designed to last long in the stomach, and they release the drug so drug can be absorbed from the stomach. Then we have radioactive microspheres, which are basically also known as glass microspheres. They emit a beta or gamma radiation either individually or in combination with a radioactive substance. Then we have polymeric microspheres, which are composed of polymers, and they are mostly biodegradable, and they contain biodegradable polymers which prolongs the residence time when they get in touch with the fluid or mucous membrane due to its high degree of swelling nature when it comes in contact with the aqueous medium. So when they swell, their buoyancy, their density becomes low, and they start floating. So this also results in the formation of gel. The concentration of polymer in the microsphere, it basically responsible for the control rate and amount of the drug which is released, which is also constant from the matrix. Now this depends upon the concentration of polymer and the release order in a persistent way. Synthetic polymer microspheres are those which are made up of synthetic polymers and are used as bulking agent, fillers, embolic particles, drug delivery vehicles, etc. Now talking about advantage of these systems, they are basically a very effective delivery of insoluble or sparingly soluble drugs. They help in masking of bitter taste, masking of any foul odor. They can also improve the viability of various drugs by slowly reducing the drug which is poorly soluble. They are used to deliver or co-deliver more than one therapeutic agent which can be suspended in the microsphere matrix. They are also used for targeting of drugs to a specific site, either in the GIT, like you can target stomach, you can target a small intestine, you can target large intestine. They are also helpful for the reduction in dose by control release or toxicity of various drugs, because when dose is reduced, side effects or unwanted side effects are also reduced. Then they can be used as a carrier for drugs, they can be used as a carrier for vaccines and diagnostic agents, and they are also used in surgical procedures. They are used to produce amorphous drugs with desirable physical properties. Now other advantages are they also reduce the local side effects like GI irritation, because the irritating drug is encapsulated in a neutral coating, and so it is not coming in contact with the gastric mucosa. When it is not coming in contact with the gastric mucosa, irritation of the GI mucosa will not be there. So drugs like NSAIDs, aspirin, they can be given in the form of microspheres, from where it is slowly released. So drug dumping is not there, so there will not be any irritation, there will not be any damage to the gastric mucosa. They also provide prolonged release and lesser amount of drug to maintain the plasma concentration, and when plasma concentration is maintained uniformly, the patient compliance will be better, because when we maintain the plasma concentration, the frequency of dosing or time, how many times you are giving the drug is also reduced, which will obviously it will improve the patient compliance. Then you can also give them by parenteral route, and they have a benefit of controlling high concentration of water soluble drugs without any osmotic effect at the site of interest or at the site of release. Now about the limitation of microsphere technique, it is that in this case large manufacturing is a problem. It is not easy to scale up the microencapsulation technique. Then during the production, sometimes if your drug is of protein or peptide in nature, it can get inactivated because of the production process. Then the release rate which is designed or developed at the research stage, sometimes it is not reproducible, so release rate is also becomes poor. The cost of the materials and processing is substantially higher than those which are used in the normal formulations like tablets, capsules. So when you are designing a microsphere, now in different methods of preparation which I have discussed, you can see in the table we have three major techniques like physiochemical processes, chemical processes and mechanical processes. In physicochemical processes, we have co-servation phase separation technique, in which is further classified as aqueous phase separation technique and organic phase separation technique. You can understand aqueous means we are using water, and organic phase we are not using water, we are using a organic solvent. In aqueous phase technique, we have basically two techniques, simple co-servation and complex co-servation. While in organic phase separation method, we have emulsification, solvent evaporation, and also melt emulsification, in which we are using temperature to solidify or to melt the polymer. Now in chemical process, it can be further classified as interfacial polymerization, centrifugal force and submerged nozzle process polymerization, and in situ polymerization or inotropic gelation. While in mechanical process, we have spray drying, spray congealing or spray chilling, fluidized bed coating, we have extrusion spheronization, we have centrifugal extrusion, we have rotational suspension separation granulation, and also we have nanosizing or milling. So these are all comes under mechanical processes. In the physicochemical process or technique of physicochemical method, the microspheres are prepared by using physicochemical parameters of the drug and polymer. So we can play with those physicochemical parameters and control their size, control their properties. The physicochemical properties like solubility of the polymer, melting point, temperature, etc. This process includes physicochemical reaction, which is produced by the addition of evaporation of organic solvent or addition of aqueous phase. It contains phase separation co-servation techniques, which further divide into two different techniques like simple and complex co-servation method. The second method is organic phase separation, which contains emulsification and melt emulsification technique. In the melt emulsification technique, we use a melting point or melting of the polymer for controlling or for encapsulating the drug molecules inside the matrix. Now let's talk about solvent evaporation technique. In this technique, three phases are present, and they are core, coat and liquid manufacturing vehicle, which is known as LMV. The coating material is dissolved in a single volatile solvent, which is not soluble in LMV phase, and a core material to be encapsulated, to be dissolved or to be dispersed in a coating polymer solution. With agitation or strong agitation, this mixture is dispersed in the liquid manufacturing vehicle phase to obtain the appropriate size microcapsules. The mixture is then heated to evaporate the solvent for the polymer. The coat material is stirring around the core material and encapsulate the core. Now if the core material is dissolved in the coating polymer solution, matrix type microcapsules are formed, which are known as microspheres. The core material may either be water soluble or water insoluble. Solvent evaporation method is classified mainly into two classes. One is single emulsification technique and second is double emulsification technique. Now in single emulsification technique, like when we are preparing water and emulsion, water and oil emulsification process was developed for the encapsulation of highly water soluble drugs. In this technique, polymer and a drug contained in a polar solvent such as acetonitrile, it is emulsified into a emulsible lipophilic phase with light mineral oil commonly being used in the presence of an oil soluble surfactant such as a span. However, an important drawback of using oil external phase is cleaning of the final product, which is difficult in case of production. So this is about water and oil emulsion technique. Now in case of oil and water emulsion technique, emulsions are more widely used than water and oil emulsions because the simplicity, because of the process and easy to clean after making the product. Now in this process, both the drug and the polymer are basically dissolved. Now in general, solvent evaporation method is particularly suitable for the microencapsulation of the lipophilic drugs that can be either dispersed or dissolved in the dispersed phase of a volatile solvent. Like clonazepam represents an interesting example of a lipophilic drug whose solubility properties make it difficult to form drug-loaded microspheres by the solvent evaporation process. Now in this, you can see in the figure, in the animation which is running, the water and oil single emulsification technique, in which we are showing you single emulsification technique. You can see the dispersion in organic phase, oil or in chloroform, then it is emulsified to form a emulsion, which is a water and oil emulsion, and then it is by using formaldehyde or glutaldehyde or butanol, it is removed. We remove the aqueous phase and we get the isolated microcapsules or microparticles or microspheres. So first we have dissolved the aqueous solution and suspension of polymer, which is emulsified to form water and oil emulsion, and then it is dried by washing and by drying in a glutaldehyde or formaldehyde phase. So this is how we are harvesting our microcapsules from the technique. Now in this animation you can see the process of oil in water single emulsification technique. In this case you see the organic phase of the polymer, in which polymer is dissolved in an organic solvent, which can be acetonitrile, which can be chloroform, it can be acetone, and then it is emulsified to form a oil in water emulsion by adding the polymer with drug in the solution form. After formation of the microcapsules, they are rigidized by changing the temperature or by changing their pH, and then they are washed to remove the excess of solvent, and then they are isolated by using formaldehyde, glutaldehyde to remove the excess of the solvent. So these microspheres either they are dried by freeze drying or spray drying or tray drying, whichever technique is available. So this is the process how your oil in water single emulsification technique is used to produce the microspheres. Now the last technique of solvent evaporation method is multiple emulsion technique or double emulsion technique. And this technique is basically appropriate for the efficient incorporation of water soluble peptides, proteins and other macromolecules. The polymers are dissolved in an organic solvent, and then it is emulsified into an aqueous solution containing emulsifier to produce multiple water and oil in water dispersion. The organic phase acts as a barrier between the two aqueous compartments, preventing the diffusion of the active material towards the external aqueous phase. So we can have two materials dispersed in two different phases, and we can prevent their interaction. Now microspheres manufactured by the water and oil in water method exhibit various morphologies such as porous or non-porous external structure depending upon the different parameters. They are classified into basically three different types of techniques: water and oil in oil double emulsion solvent evaporation technique, water and oil in water double emulsion solvent evaporation technique, and water and oil in water double emulsion solvent extraction technique. So there are three different techniques which are used in this method. Now in multiple emulsion technique, when we are preparing water and oil double emulsion solvent evaporation technique, in this method the internal aqueous phase and external oil phase are separated by another oil phase. In the water and oil method, hydrophilic compounds are first dissolved in the aqueous phase. Then it is emulsified to form a water and oil emulsion, which is a primary stable emulsion. This primary emulsion is further dispersed into a solution of polymer, which is dissolved in an organic solvent to form a water and oil in oil emulsion. Hydrophilic compounds cannot be diffused into the medium since the oil is outer dispersing medium. The total encapsulation efficiency is high as compared to other technique for a hydrophilic drug, because hydrophilic drug mostly leaches out when it comes in contact with the aqueous phase, but because it is encapsulated by a oily phase, it will not leach out and it will really remain encapsulated in the emulsion. Now in multiple emulsion technique, when we are preparing water and oil double emulsion solvent evaporation technique, in this method the internal aqueous phase and external oil phase are separated by another oil phase. In the water and oil method, hydrophilic compounds are first dissolved in the aqueous phase. Then it is emulsified to form a water and oil emulsion, which is a primary stable emulsion. This primary emulsion is further dispersed into a solution of polymer, which is dissolved in an organic solvent to form a water and oil in oil emulsion. Hydrophilic compounds cannot be diffused into the medium since the oil is outer dispersing medium. The total encapsulation efficiency is high as compared to other technique for a hydrophilic drug, because hydrophilic drug mostly leaches out when it comes in contact with the aqueous phase, but because it is encapsulated by a oily phase, it will not leach out and it will really remain encapsulated in the emulsion. Now in multiple emulsion technique, where we are talking about water and oil in water double emulsion solvent evaporation technique, in this an aqueous solution or suspension of the drug where internal aqueous phase is W1, this is emulsified in a solution of polymer which is dissolved in an organic solvent. The resulting prime emulsion, which is now water and oil, is then diffused in a second aqueous phase which is W2, containing a suitable emulsifier or a stabilizer to form a water and oil in water emulsion. So it becomes a multiple emulsion. Now evaporation of the volatile organic solvent, it leads to the precipitation of the polymer, and which leads to the formation of solid microparticles. The solid matter particles, they are separated by centrifugation, and then after washing it with a proper solvent, the main purpose of washing is to reduce the residual emulsifier and dry it under vacuum. The organic solvent which is used to prepare the microparticles by the double emulsion solvent evaporation method, it should be of low boiling point to facilitate the reduction of residual solvent by drying method at a lower temperature. Because this residual solvent sometimes it can be toxic, so it has to be completely removed from the microparticles. Now various solvents that can be used for the preparation of microparticles are ethyl acetate, chloroform, acetonitrile, benzene and methylene chloride. Although they are being toxic and they are under controlled category, but we control the amount of solvents by drying, and also we measure the amount of solvent as a residual solvent by any technique like HPLC or GLC. Now in water and oil in water double emulsification technique, the solvent can be removed from the microparticles by extraction of the solvent present in the internal phase. This can be performed by the addition of a water and oil in water emulsion to a third solution, which is a non-solvent of the polymer but miscible with both aqueous and organic solvent. Under these conditions, the solvent contained in the polymer droplets is extracted into the external aqueous medium. In this technique, an aqueous solution or suspension of the drug is emulsified in a solution of polymer in an organic solvent. The resultant primary emulsion is then dispersed in a second aqueous phase having suitable emulsifier or stabilizer to form a double emulsion. Now removal of this solvent by extraction resulted in the formation of solid microparticles. Now the process variables which are important in the solvent evaporation technique is solubility of polymer in the organic solvent, solubility of the organic solvent in water, concentration of the polymer, ratio of dispersed phase to continuous phase which is known as DPCP ratio, the rate of solvent removal, interaction between drug and the polymer, solubility of drug in the continuous phase, molecular weight of the polymer, effect of solubility of the drug in continuous phase, and what is the rate of shear or what is the rate of agitation which is being used for the dispersion of the polymer or the phases when two phases are mixed. So these are the process variables which are very important, and when developing a formulation or method or process for the preparation of microspheres, all these variables should be remain under control and should be considered before designing this process. Now in the end, let's talk about the advantages and disadvantages of solvent evaporation technique. It is a most widely used technique because of its simplicity and easy to perform. In this technique, combination of emulsifiers increases the stability of the multiple emulsions for long time storage. It means its shelf life is much higher than other techniques. It also allows incorporation of both types of drugs, hydrophilic as well as hydrophilic drugs, and it also shows very good reproducibility. It is very easy handle. Now disadvantage which is there are preparation needs to be shaken well before use because sometimes it is not properly distributed. A measuring device is required for the administration. Then a degree of technical accuracy is also required to measure the dose. Storage condition may affect the stability, bulky, difficult to transport and prone to container breakages. It is liable to microbial contamination which can lead to cracking. So all these problems has to be keep in mind when we are designing the final formulation. So these problems should not come up and should not affect the stability or quality of the product. Now in the melt emulsification technique, which is also known as melt solidification, or this is also called as emulsion congealing or hot emulsion or melt dispersion or hot melt microencapsulation. So this technique uses lipids, waxes and polyethylene glycols. They are the most favorable carriers for making these types of microcapsules. Mostly lipophilic drugs are suitable for these methods due to their hydrophobic nature. Their drug is which is incorporated in these carriers to achieve the controlled release, taste marking and stability improvement. It mainly comprises of a lipid carrier, a hydrophobic drug and a stabilizer. So all these three are required to form a microspheres using melt emulsification technique. Now in the melt emulsification technique, the molten drug and wax mass is emulsified in a aqueous phase. Aqueous phase can be a water or it can be any buffer having a temperature which is higher than the melting point of the wax. Then it is followed by its solidification by either chilling or by cooling the entire mass. Now usually in this method we have two water paths. One have high temperature, which is greater than the melting point of the wax. Another have low temperature. So in the first process, the emulsification of molten drug wax mass happen. Then when we pour to another path where the solidification of drug wax mass happens because of cooling. So when it is solidified, it becomes a solid mass in the form of a microsphere. Now advantages of melt emulsification techniques are: it is not a complicated technique, it only requires control of temperature. There is no use a organic solvent, so it is a solvent free method, which obviously make it a very attractive method where we are not using any solvent. So in those conditions where solvent is a completely banned, we can use this technique. By this method we can also improve the sustained release properties for some formulation, especially for drugs which cannot be designed by other techniques. In this case dose dumping is not there. Then it is basically low melting points are also avoided. It shows good stability at a varying pH and at a varying moisture level because it is encapsulated in a hydrophobic carrier. It is to be used in humans and it is safe because of their non-soluble and water insoluble nature. So it is much more safer than other types of products which are produced by other techniques or other emulsification techniques. So students, let's now summarize what we have learned from this module. In this we have learned what are microspheres, different types of microspheres, what is their composition, what are their merits, demerits, like they provide protection for unstable drugs before and after the administration prior to their availability to the site of action. They can also control and at the rate of drug release at a predictable rate with a smaller dose of the drugs. They also improve patient compliance, especially for those drugs which are basically for chronic indication, or especially those drugs where frequency of dosing is very high or half-life of the drug is low, so it has to be given to the patient many times a day. But if you design a controlled microsphere, in that case you can reduce the dosing and you can improve the bioavailability of the drug and you can improve the patient compliance. Secondly, microspheres, because they are free-flowing dry powders, they can be easily filled into capsules, they can be filled into bottles, they can be filled into in the form of dry syrup. So they can be given in various forms because they are of their free-flowing property and dry property. They can be used to convert semi-solids or liquids into dry free-flowing powders. Thank you.